Good morning, children. Yesterday we started with the process of translation, and I told you the first three steps we have already discussed, and those three steps which we have already discussed are activation of amino acids. The first step which we have discussed is activation of amino acids. Then the second step which we have discussed is charging of the RNA. This also we have discussed. And the third step, which we have discussed yesterday, is activation of ribosomes. So we have already discussed these three steps, which are there in the process of translation. So today we are going to discuss now the fourth step, and the fourth step is initiation of polypeptide chain. Polypeptide chain starting, the initiation of formation of polypeptide chain takes place. Now this basically takes place in ribosomes, and during which what happens that the messenger RNA, which brings the coded message, it attaches to smaller subunit and is decoded by the smaller subunit. Then, as you know, the larger subunit has three sites. Site. So, beta, this connects to the smaller subunit by the 5' end. Now what happens? The ribosomes, as you know, the ribosomes have three sites, and the first codon, which is read by the ribosome, the first codon, which is read by the ribosome, is the initiator codon. and that initiator codon is initiation codon, which starts the process of formation of polypeptide chain is AUG. This is the first codon which initiates the process of formation of polypeptide chain. So this will be the first codon which will be read by the messenger RNA. And then accordingly, the tRNA will carry the amino acid which codes for AUG. That means the amino acid which codes for AUG is methionine. That means a transfer RNA which will have it will have anticodon for the initiator codon which is there on messenger RNA. This is initiator codon on the 5' end, which is AUG. AUG. So it will have the transfer RNA will have the anticodon for this, and that will be UAC. And here to the OH group, 3' end and OH, where OH functional group is present, it will carry the amino acid which codes for AUG, and that is methionine. For AUG, methionine. So it will carry methionine. It will attach to methionine, and methionine will be carried by this transfer RNA. And where it will be? It will be carried here to the larger subunit. And which particular part of the larger subunit? The initiator codon. When the formation of polypeptide chain begins, the initiator codon is the first one which is decoded is X site. Anybody who remembers? The messenger RNA, the amino acid which is brought by this initiator codon, by this transfer RNA, it is received by which site? There are 3 sites on larger subunit of ribosome, ribosome. So which particular site receives this transfer RNA which is carrying amino acid for initiator codon? Can you tell me? When it starts the first codon, which is initiator codon, the transfer RNA which carries amino acid for this initiator codon, which site receives this transfer RNA? Which site receives this transfer RNA? Which site of larger subunit receives this transfer RNA? Anybody who can answer this question? Yes, beta. Which site of larger subunit receives this transfer RNA, which is bringing methionine for the initiator codon? Yes, anybody who can answer this question? Yes, beta. Sukhsahaj, P site. That's right, Sukhsahaj. Very good. It is the P site. That means when the initiation of polypeptide chain starts, the first codon, which is red, all the messenger RNA will have AUG as their first codon, and that is why it is known as initiator codon. The rest of the codons will be different, but this initiator, the starting codon, the first codon on the 5' end will be initiator codon, and this initiator codon codes for methionine, which will be carried by a transfer RNA. So when this transfer RNA comes, it will be accepted by the P site. It will be accepted by the P site. There are 3 sites on the larger subunit, the E site, the P site, and the A site. So this transfer RNA, which will carry methionine, will be accepted by P site. This is E site and A site. So it will be accepted by P site. P site accepts the first amino acid which is carried by a transfer RNA. Rest then as I told you before also, rest of the triplet codons are different in different messenger RNA. But this is same in all because this is the initiator codon and it is accepted by P site. Now suppose the next is UUU. This UUU codes for the amino acid phenylalanine. Phenylalanine. Rest of all, that means the transfer RNA for this codon will bring the transfer RNA for this codon will bring phenylalanine. Okay, and which site will accept? Which site will accept this transfer RNA? E site, P site, or A site? Which site will accept this? Yes, A site. That's right. Very good, Suksahaj. So after the initiator codon, all the these triplet codons for which the amino acid will be brought are accepted by A site. Only the initiator codon, only for the initiator codon, the transfer RNA is accepted by P site. Rest whatever number of triplet codon is there. That means suppose we have here, say GUG, it codes for valine. So valine, the messenger RNA which brings valine, that will be accepted by this. So rest all are accepted by A site. A site accepts rest all the transfer RNA, bringing the respective amino acids. Only the initiator codon, only the RNA, the tRNA, which is bringing amino acid for the initiator codon is accepted by P site. The first triplet codon, which is AUG, for which the transfer RNA, whatever that means AUG codes for methionine. So the transfer RNA which brings methionine, the first initiator codon, the amino acid for the initiator codon is accepted by P site. Rest all are accepted by A site. That means the next is UUU, which codes for phenylalanine. That means now one transfer RNA will bring phenylalanine because the next code is, next triplet codon is UUU. UUU codes for phenylalanine. That means a transfer RNA is required which will carry phenylalanine. And when this comes near it, it will be accepted by A site. A site will accept. Then when A site accepts this, that means we have in peptidyl side methionine, which is for initiator codon AUG, and on A side we have transfer RNA with phenylalanine, which codes for UUU. Now what happens? We have one here and one transfer RNA here, and both of these transfer RNA with their respective amino acids are in the larger subunit. This is the larger subunit. Both are in the larger subunit of ribosome. Both are in the larger subunit of ribosome. Now what happens? I told you there is an enzyme which is present in larger subunit. Can you name that enzyme? Name the enzyme which is present in larger subunit of ribosome. Yes, what is that enzyme? Yesterday only I discussed with you. Yes, name the enzyme which is present on larger subunit of messenger of ribosome. Can you name that enzyme? Yes, beta. Name the enzyme which is present in the larger subunit of ribosome. Who can name the enzyme? Yes, Suksahaj, amino acid tRNA synthesis. No, beta, Suksahaj, this is required for activation of amino acids and charging of tRNA. The enzyme which is present in the larger subunit of ribosomes is peptidyl transferase. The enzyme which is present in larger subunit of ribosome is peptidyl transferase. And I have told you it has two functions. Yesterday also I told you, today I am repeating it again. Number one function of this is formation of peptide bonds. It forms the peptide bonds in between the amino acid. And the second is termination of polypeptide chain. Two functions. When the polypeptide chain is formed completely, it is terminated or it is cut from the whole setup. So that is also brought about by this. Now, B site has a amino acid for the initiator codon, and A site has received the amino acid for the next codon, which is UUU. So one methionine and one phenylalanine are there, two amino acids, which are carried by these clover leaf-like structures, which are known as transfer RNA. Who's carrying these amino acids? Transfer RNA is carrying these amino acids. Now what happens? Because of the, see, these, if the amino acids are kept like this, methyl, methionine, phenylalanine, these are of no use. They are just amino acids. They are just amino acids. If they are separate, they are just amino acid. What makes them a protein molecule? When they are joined by peptide bonds, then it forms a protein molecule. Now, that means the methionine and phenylalanine, which is carried by transfer RNA, they need to be connected, joined by peptide bonds. So as I told you, in this larger subunit, an enzyme peptidyl transferase is present, which forms the peptide bond. So with the help of this enzyme, which is present in the same subunit, a peptide bond is formed between these two. A peptide bond is formed between these two amino acids, that means methionine and phenylalanine. This peptide bond has connected the methionine as well as phenylalanine. That means the amino acids have started converting into protein molecule or polypeptide chain. Now what happens? Now after this, already this has been started. Now elongation. The next step which takes place here is, I'll erase this. The next step which takes place here is, make it small, which takes place here is the fifth step, which is elongation of polypeptide chain. Elongation of polypeptide chain. That means right now only two have joined together. That means methionine has joined with phenylalanine, and we have got methionine, this amino acid, connected to another amino acid, phenylalanine. This makes a very small polypeptide chain, and the polypeptide chain needs to be long, so that, or whatever length, on the basis of decoding the message which is present in messenger RNA, whatever length of triplet codons are present on messenger RNA, on that basis, the length of the protein molecule or polypeptide chain is determined. Now here, till now two have come, so both are connected by this. Now it has to be elongated. That means more of the transfer RNA bringing more of the amino acids are required, so that this chain elongates. So now elongation takes place. Now what happens? Elongation starts. See, elongation. See, first what has happened? The anticodon on this on the transfer RNA read this message and then brought the amino acid, respective amino acids. The second thing which happened that elongation. For elongation, what was required? Formation of peptide bond between the amino acids. This also has happened. Now what happened? The third step involves translocation. See, first step, this, the code is read by anticodon of transfer RNA. Second step, formation of peptide bond between the amino acid. Now the third step, I'm writing here, first is anticodon of transfer RNA binds with. So anticodon reads the codon on messenger RNA. This happened as the first step. A, I'm writing. Then B, during B, peptide bond formation. Peptide bond formation between amino acids took place. And now the third thing, which is very important, see, the third important thing which takes place during the elongation is translocation. Translocation. Translocation is very important. What is this translocation? Translocation is, it's not, see, this one is read by the respective transfer RNA. This is also read. Now it is the turn of the next triplet codon to be read by messenger RNA, and then the fourth codon will be read by messenger RNA. Suppose it is again U. Then this way the fifth will be read by the messenger RNA. Suppose it is UAG. No, this is a stop codon. So let it be GAG. Okay, this will be read. Now for that, these should come here, here. Below this, they should be shifted here. These should come down the side. This should move along so that this goes on this side. These two which are already red, it shifts this side, and these which are not red, they come into this side. So for that, what is required is translocation. Now, if we say what is translocation? Translocation is the movement of ribosomes along messenger RNA. What moves? It's not that it will move. This moves. The ribosome, it moves. That means it has read this, it has read this. Now it will shift here, the whole ribosome. It will shift here to this now. This ribosome, the larger subunit and the smaller subunit, it will shift here now. That means from here, the ribosome will come on GUG and UUU. This whole, the larger subunit and the smaller subunit will shift from here to here. And this process of movement of, I'm writing here, movement of ribosomes on messenger RNA, movement of ribosomes on messenger RNA, movement of ribosome along messenger RNA, along messenger RNA is known as translocation. And what does it require? It requires a form of energy, and that energy is GTP, guanosine triphosphate, plus one factor is also required, and elongation factor is also required, and that elongation factor is also required, and that elongation factor is EFG factor. EFG factor. So under the influence of the energy by GTP and the factor EFG factor, what happens? This ribosome shifts from, because two are read now, from here to here, so that it comes over GUG and UUG. It shifts from here to here. And this process is known as translocation. So when translocation takes place because of the movement of ribosome, what will happen? Some change will also take place. And what is that change? So during translocation, when this ribosome shifts from here to here, this transfer RNA, which is at P site, it with a jerk, because a jerk takes place, with a jerk, this methionine comes to here, P site, and this phenylalanine comes to this side. That means A site is again empty. This is joined by this, both are joined by this bond, peptide bond. So now that means they have shifted because of translocation. Now A site is again empty. And now this ribosome has come on GUG. GUG codes for valine. So now what is required? Valine. So another transfer RNA with valine will be accepted by acceptor site. Now what will happen? This has received for the next one. So now peptidyl transferase will form a bond here. Okay, bond is formed between two. And again, there will be translocation. Again, translocation will take place. Now this time, because of translation, again, everything will shift. How shifting will take place now? This time when translocation takes place, how dislocation will take place? The dislocation will be the thing which is at E will now go out. That means methionine will move out, and it will get separated from its transfer RNA. So it will get separated from its transfer RNA and it goes out. And this phenylalanine, where will it come? It will come in the place of E. Phenylalanine has come here. And this will now, valine will come here. This is also joined by this. And this is again empty. This is again empty. Now this is ready to receive another transfer RNA. Now what is the next one? It is U again. Now it has shifted more. It has shifted to U now because of that shifting only, because of that translocation only. Everything has moved here. Now this is again empty. Now when it is empty, now which next codon is red? UUU is red. UUU codes for phenylalanine. So another transfer RNA will come, which will bring another phenylalanine. Now again, they are close, and here peptidyl transferase is there. So a bond will be formed here. And then that means we have now a long chain of amino acids. Phenylalanine, then we have valine, and then we have again phenylalanine. We have again phenylalanine. Then again, what will happen? Translocation will take place. Now, this time when this ribosome moves on to next GAG, it moves on to here, more shifting will take place. That means this time this P will also go out. This will move up. So methionine will move up. Methionine has moved up. This P will also move out. Phenylalanine will move out. And this V will come, shift to the E side. Okay, and this phenylalanine has shifted here, and again it is empty to receive another amino acid. This is again empty to receive another amino acid. So the next amino acid will be received, and this way elongation of peptide chain will take place. Is it clear, beta? Anything you want me to repeat in this, or is it clear? Yes, bache? Is it clear? Yes. Anything you want me to repeat in this, or this is clear? Anything which you could not understand? Just I want your response, bache. Is it clear? Yes, beta. Sukhsehaj is answering. Any other? I want more response. Is it clear this process? Then the last step is left. I'll tell you that. Yes, beta. I have got just one response. Chitra is also saying yes. Others, please respond quickly. I want more responses. Only 2 students are saying that it is clear. You can tell me the point from where I need to repeat this process. Yes, how many students are there? Yes, Mariam, is it clear? Send me the message. Is it clear, Mariam? Vanithi, Shagun, Shruti Lakade, is it clear this process? Mariam, Vanithi, Shagun, Shruti Lakade. Any point you want me to repeat? I'm not getting your response, beta. Only Shruti Lakade has responded. She's saying it's clear. Okay, Navneet, Aditi Gurang, Chirag, Girish, is it clear? Aditi Gurang, Chirag, Girish, Arif, is it clear? This is a very important process. That's why I'm asking you all. Aditi Gurang, Chirag, Girish, Arif. Is it clear to all of you? This is a very important process. Yes, I am not getting your response, beta. Yes, Arif? You want me to repeat it? Yes, bache? Chirag? It needs to be repeated. You can ask in the form of questions also. If you do not understand, ask me questions. I'll reply you. uh, you will be able to understand what you could not understand if you could not. Yes, Girish? Yes, Girish? Am I audible to you? Arif, am I audible? No response from any of you. Shall I repeat it? Yes, Arif is answering. Okay, Arif is saying that it's clear. Girish, I don't know why you are not answering. At least answer that it is clear, or if not clear, just let me know the points which are not clear. Parthavi, Simran, Sukh Sahaj already answered my question. Parthavi, Simran, Suzanne. Parthavi, Simran, Suzanne. I have unmuted you. Now you let me know. I have unmuted you all. Just let me know. Yes. Chirag, Girish, Aditi, Arif. Arif has answered. Parthavi, Simran. Okay, Parthavi, Suzanne, when you will upload yesterday's lecture? Yesterday's lecture I have already uploaded. Parthavi, it's been uploaded. It's there. And I have shared also in the group. And I have shared a sample paper also. Sukh Sahaj, have you seen that sample paper? The sample paper provided by CBSE, that also I have uploaded. Okay, check check it. This is a sample paper, quite nice, easy. Check the sample paper also, see the sample paper also, that also has been uploaded. I have sent that in the group. And every day I upload the lecture every day. You need to check the bio group, and plus in uh, I send it in the bio group, and I send it in 11th and 12th group. 12th science. So it's already been uploaded. But what matters is this process of translation till here clear to you? This is what I want to ask. Chirag, is it clear? Chirag, you are unmute. You can speak up. Is it clear, beta? Girish, is it clear? Mom, I don't know whether you all are sitting in front of your laptops and mobiles or not, because you people are very quiet. You people are not answering. Yes, bache? Girish? Mariam Vanithi Shagun? Mariam Vanithi Shagun? Shagun has answered. Clear.